Welcome to the lecture on endocrine system. In this lecture, I will describe the function of endocrine system. I will talk about hormone, how hormone is regulated, and then I will also discuss major endocrine glands in our body. Glands are made of epithelial tissue, and there are two types of glands, endocrine gland and exocrine gland. Exocrine glands are the type of glands in which the gland releases its product on its surface, such as skin or the inner lining of hollow organs. Exocrine gland has a duct which direct the secretion or the product of the gland to the surface. On the other hand, we have endocrine gland. They do not have a duct. Instead, they release their product directly into body fluid such as blood. Hence, these glands are also called ductless gland. The product of endocrine gland is called hormone. And hormones are released in the blood and they travel through blood, or they are transported through blood to distant part of the body and act on specific cells. Those specific cells which has the receptors for specific type of hormones are called target site or target cells. Endocrine cells are distributed throughout the body. However, in certain areas of our body, we have a relatively larger accumulation of these cells, and they constitute major endocrine glands. They're located in different parts of our body. For example, we have them in the brain. There are endocrine glands in thoracic cavity, also in abdominal cavity and pelvic cavity. The overall function of nervous system is to maintain homeostasis. This is similar to one of the main functions of the nervous system, which is also to regulate homeostasis. Hence, we can make a contrast and comparison between these two systems. So to start with, we say that the type of cell in the nervous system is a neuron. However, in the endocrine system is epithelial tissue. In the nervous system, neurons communicate with another neurons or with a muscle in synapse through a release of chemicals called the neurotransmitters. However, in the endocrine systems, the endocrine gland cells communicate with target cells by release of chemicals called hormone. The onset of action in the nervous system is quite rapid compared to the endocrine system. Similarly, the duration of action is quite short in the nervous system compared to endocrine system. Structurally speaking, hormones are divided into two categories. There are some hormones which are made of steroids, so we call them steroid hormones, while there are other hormones which are not made of steroids. For example, they are made of protein, glycoprotein, or what have you, so we call them non-steroid hormones. Non-steroid hormones act on receptors located on cell membrane. However, steroid hormones can cross cell membrane and they act on receptors located either in the cytoplasm or inside the nucleus.
Hormonal communication generally begins with a part of the neuroendocrine system receiving sensory information and reacting by issuing a command to the body in the form of a hormone. In this example, dehydration is detected by osmoreceptors in the hypothalamus, which then directs or stimulates the posterior pituitary gland to release antidiuretic hormone, ADH. The next step is to transport the hormone molecules to target cells. This is typically done via the bloodstream. Here, ADH is transported through the bloodstream to the kidneys and blood vessels. When hormone molecules reach target cells, they will bind to matching receptors on those cells, and the hormone receptor complexes will trigger changes in the target cells. This causes the kidneys to reduce the urine volume output, thereby increasing water retention and countering dehydration. In blood vessels, vasoconstriction is increased, leading to higher blood pressure and thus countering the blood pressure drop caused by dehydration. The membrane-bound receptor has a receptor site on the outside of the cell for a small signaling molecule called a ligand. The portion of the receptor on the inside of the cell can bind to the G protein, which consists of three subunits, gamma, beta, and alpha. A guanosine diphosphate is attached to the alpha subunit. When the ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation and separates from the membrane-bound receptor. Guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from the receptor site, additional G proteins are no longer activated. Inactivation of the alpha subunit occurs when its own phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunits then recombine and attach to the receptor in the cell membrane.
Second messengers, or intracellular mediators, effect changes inside of cells when a signal molecule binds to a receptor on the surface of the cell's plasma membrane. The binding of the signal molecule, the first messenger, to its receptor activates a nearby G protein. Depending on the target cell, the effect of the G protein varies, typically activating one of two different second messenger pathways. Some second messenger systems involve activating the production of cyclic AMP. When a signal molecule, such as the hormone epinephrine, binds to a cell surface receptor, it activates a G protein on the inside of the cell membrane. The G protein then stimulates adenylate cyclase to produce large amounts of cyclic AMP within the cell. The cyclic AMP then binds to and activates a target protein such as alpha kinase, which adds phosphates to specific proteins in the cell. The effect of activating cyclic AMP is a cascade, an amplification, as each molecule of cyclic AMP activates many enzyme molecules, each of which activates many more molecules. Multiple copies of activated alpha kinase have phosphorylated even more molecules of some specific protein. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated. Calcium ions are involved in other second messenger systems. In response to a signal molecule from other cells, the cell surface receptor activates a G protein, which in this case activates the enzyme phospholipase C. This enzyme catalyzes the production of inositol triphosphate, or IP3, which diffuses through the cytoplasm to the endoplasmic reticulum. The IP3 binds to and opens the calcium channels, allowing calcium ions to flow from the endoplasmic reticulum into the cytoplasm. The calcium ions trigger proteins sensitive to calcium to initiate a variety of activities. Whether triggering phosphorylation or the release of calcium ions, second messenger systems, or intracellular mediators, allow signal molecules attaching to the outside of the cell to have an effect on activity inside the cell.
Aldosterone is a lipid-soluble hormone that can easily diffuse through the plasma membrane. Inside the cell, aldosterone binds with an aldosterone receptor molecule in the cytoplasm. The aldosterone receptor complex moves into the nucleus and binds to DNA. The binding of the aldosterone receptor complex to DNA stimulates the synthesis of messenger RNA, which codes for specific proteins. The messenger RNA moves from the nucleus into the cytoplasm and binds to ribosomes, where it directs the synthesis of specific proteins. These proteins produce the response of the cell to aldosterone.
Generally speaking, the endocrine system is maintained by a negative feedback mechanism. And there are three ways through which negative feedback mechanism is achieved. Number one is through hormone. So for example, one hormone from a gland will go ahead act on another gland. It will either stimulate or inhibit the secondary gland. Number two, through nervous system. So the autonomic division of the nervous system will either stimulate or inhibit release of hormone from a gland. Number three, by blood level of a chemical substance. For example, blood sugar level will either act as a stimulus or as an inhibitory factor on pancreas to release or inhibit pancreas hormone.
The hypothalamus is sometimes referred to as the master control center of the endocrine system because it regulates most endocrine functions of the body. Located at the base of the brain in the anterior portion of the diencephalon, the hypothalamus consists of several groups of neurons called nuclei, each with specific functions. The stalk-like infundibulum connects the hypothalamus to the pituitary gland or hypophysis. The pituitary gland sits within the hypophyseal fossa of the sphenoid bone and is divided into anterior and posterior lobes. Each lobe of the pituitary gland interacts with the hypothalamus in a different way. The anterior pituitary, or adenohypophysis, is connected to the hypothalamus by a blood vessel network known as the hypothalamal hypophyseal portal system. Hormones released by the hypothalamus travel through the portal system and regulate activities of the anterior pituitary. In contrast, the posterior pituitary, or neurohypophysis, is connected to the hypothalamus by neurons that form the hypothalamal hypophyseal tract. Axons of these neurons transport hormones from the hypothalamus for storage in the posterior pituitary. The anterior pituitary is regulated by two classes of hypothalamic hormones, releasing hormones and inhibiting hormones. These hormones reach the anterior pituitary via the hypothalamal hypophyseal portal system and stimulate or suppress secretion of anterior pituitary hormones. Hormones secreted by the anterior pituitary then enter the bloodstream for distribution to their specific target tissues. For example, thyrotropin-releasing hormones secreted by the hypothalamus stimulates thyrotropic cells of the anterior pituitary to increase secretion of thyroid-stimulating hormone, which then regulates activity of the thyroid gland. The posterior pituitary produces no hormones of its own. Rather, neurosecretory cells in the hypothalamus synthesize two hormones, antidiuretic hormone, known as ADH or vasopressin, and oxytocin, that are transported to the posterior pituitary via the hypothalamal hypophyseal tract. These hormones are stored in nerve terminals in the posterior pituitary and released into the blood upon stimulation of the axons. Each hormone has distinct functions. ADH acts on the kidneys to increase water retention from renal tubules. It also acts to constrict blood vessels. Oxytocin causes smooth muscle contraction in both the male and female reproductive tracts. It also stimulates milk ejection by the lactating mammary glands.
The thyroid gland is the largest endocrine gland, located in the neck inferior to the larynx and anterior to the trachea. This butterfly-shaped gland has left and right lobes connected by a narrow band of thyroid tissue called the isthmus. The thyroid gland is highly vascular, giving it a reddish color. Blood reaches the thyroid via superior and inferior thyroid arteries. Superior, middle, and inferior thyroid veins carry thyroid hormones from the gland. Beneath the fibrous capsule, the thyroid gland is composed of spherical structures called thyroid follicles. The follicle wall is a simple cuboidal epithelium of follicular cells that surround a central lumen. The follicle lumen is filled with a viscous colloid that contains a glycoprotein called thyroglobulin. Capillaries and parafollicular cells are located in the loose connective tissue between thyroid follicles. Parafollicular cells, also called C cells, secrete the hormone calcitonin, which is involved in regulating calcium levels in body fluids. Calcitonin is the antagonist to parathyroid hormone. Thyroid hormone, or TH, is a general term that encompasses two specific hormones: triiodothyronine, known as T3, and tetraiodothyronine, known as T4, or thyroxine. Thyroid-stimulating hormone, or TSH, from the anterior pituitary maintains TH synthesis and secretion. In the first phase of TH production, follicular cells secrete thyroglobulin, which contains numerous tyrosine amino acids, into the follicle lumen. Iodide molecules, I-, from the blood, cross the follicular cell and enter the lumen where they are converted to iodine, I. The tyrosine molecules within the thyroglobulin combine with iodine to form iodinated thyroglobulin, which can be stored in the follicle lumen for 2 to 3 months. In the second phase, follicular cells absorb iodinated thyroglobulin by endocytosis, and lysosomal enzymes hydrolyze the thyroglobulin to produce T3 and T4, which are secreted into the bloodstream. The primary effect of TH is to maintain normal cellular metabolic activity. In children, TH is especially important for the growth and development of many tissues through stimulation of the pituitary gland to release growth hormone. Excess production of TH, hyperthyroidism, causes increased metabolic rate, which can lead to weight loss, hyperactivity, heat intolerance, and goiter, or enlargement of the thyroid. Insufficient TH production, hypothyroidism, results in low metabolic rate and resultant weight gain, sluggishness, and sensitivity to cold temperatures. The small parathyroid glands are located on the posterior surface of the thyroid gland. There are usually 4 parathyroid glands, although the number varies between 2 and 6. They are supplied by the inferior thyroid arteries. Parathyroid glands are composed of 2 cell types: chief cells, or principal cells, which synthesize parathyroid hormone, and oxyphil cells, whose function is unknown. Parathyroid hormone, or PTH, is secreted in response to low blood calcium levels. PTH raises calcium levels in several ways. It promotes resorption of calcium from bone tissue. It increases resorption of calcium in the kidneys, so it is not excreted in urine. And it stimulates synthesis in the kidney of the hormone calcitriol, the active form of vitamin D, which increases calcium absorption from the small intestine. Excess PTH secretion, hyperparathyroidism, may lead to bone fractures due to reduced calcium, kidney stones, psychological changes such as depression, muscular weakness, or even cardiac arrest if blood calcium levels are too high. Inadvertent removal of the parathyroid glands during surgery of the thyroid gland is the most common cause of hypoparathyroidism. The lack of PTH causes low blood calcium levels, which can lead to neuromuscular symptoms such as tingling in the fingers and limbs, muscle cramps, cardiac arrhythmia, and tetany, which may result in death by asphyxiation. The organs of the endocrine system secrete hormones directly into the bloodstream to assist in the regulation of various body functions. The endocrine glands discussed here are the suprarenal glands. The paired suprarenal, or adrenal glands, are pyramid-shaped endocrine glands located on the superior pole of each kidney. These glands receive a rich blood supply from multiple arteries and are drained by a single suprarenal vein. The suprarenal glands are composed of an outer cortex and an inner medulla. The cortex synthesizes more than 25 steroid hormones, known collectively as corticosteroids. The cortex consists of 3 layers. The zona glomerulosa is the thin outer layer. Its cells synthesize mineralocorticoids, a group of hormones that help regulate electrolyte composition and concentration in body fluids. The principal mineralocorticoid is aldosterone, which helps regulate sodium and potassium ions in the blood and body fluids. The zona fasciculata is the thick middle layer of the cortex. It is composed of lipid-rich cells that secrete glucocorticoids. These hormones stimulate metabolism of lipids and proteins, help regulate glucose levels in the blood, and play a role in decreasing inflammation. The most common glucocorticoids are cortisol and corticosterone. The zona reticularis is the deepest layer of the cortex. Its cells are arranged in irregular cords and produce adrenal sex hormones, including weak androgens and small amounts of estrogens. The inner core or medulla of each suprarenal gland consists of large clusters of chromaffin cells. When stimulated by sympathetic neurons of the autonomic nervous system, the medulla secretes the hormones epinephrine and norepinephrine. As these hormones circulate, they initiate multiple systemic mechanisms that contribute to the body's fight-or-flight response. The organs of the endocrine system secrete hormones directly into the bloodstream to assist in the regulation of various body functions. The endocrine gland discussed here is the pancreas. The pancreas is an elongated organ situated between the duodenum of the small intestine and the spleen, inferior and posterior to the stomach. It is both an exocrine and endocrine gland. The pancreas consists primarily of exocrine acinar cells and their associated ducts. Organized in glandular clusters, pancreatic acini secrete enzyme-rich pancreatic juice via the pancreatic ducts into the duodenum to aid chemical digestion. Scattered among the acini are small clusters of endocrine cells known as pancreatic islets, or islets of Langerhans. Each pancreatic islet contains 4 types of cells: alpha, beta, delta, and F cells that release hormones into the bloodstream. When blood glucose levels decline, alpha cells secrete glucagon, a hormone that stimulates the liver to break down stored glycogen and release glucose into the bloodstream. When blood levels of glucose and amino acids rise, such as after a meal, beta cells secrete insulin. Insulin stimulates cells to absorb glucose and amino acids from the blood and to store excess nutrients. Delta cells synthesize somatostatin. This hormone inhibits the release of insulin and glucagon and slows the activities of digestive organs, thereby slowing the entry of nutrients into the bloodstream. F cells secrete pancreatic polypeptide to suppress and regulate somatostatin secretion by delta cells. Together, these pancreatic hormones provide for the orderly uptake and processing of nutrients.